We've already talked about the plasma membrane here. We mentioned that it was sort of the security guard. It was semi-permeable and only let some things into the cell and not others. But now we want to take some time to talk about the various types of transport mechanisms that allow things in and out of the cell. As we've seen already, cells are surrounded by extracellular fluid. Within that fluid, there are nutrients, regulatory substances like hormones, electrolytes, wastes. Inside the cell, there's also fluid with some of those same things. And so cells will need to move things into themselves that they want, like the nutrients, the electrolytes, water, and they need to be able to get rid of the things they don't, like wastes. And so the two main categories of transport are passive transport and active transport. Passive transport is when you have high concentration of one chemical on one side of the plasma membrane and it moves to the other side of the plasma membrane that has a lower concentration of that chemical. Ultimately, no energy is required. This process depends on molecular size, the speed of the molecules, and the temperature. But overall, again, it is a chemical moving from high concentration to low concentration and no energy is required. The analogy for that is like a ball rolling down a hill. There's already a certain amount of energy to that, and then once let go, the ball will roll down the hill from a high level to a lower level. It's a passive transport mechanism. The opposite then is the case for the active transport mechanism. To actively move that ball up the hill, it would require energy. In this case, ATP, as we mentioned, being the major form of energy in our cells. In this case, we're moving the chemical the opposite way, from a low concentration to a high concentration across the plasma membrane. Not only does it require energy, but it also requires a carrier molecule. So the person in this image here is representing a carrier molecule, a protein that helps the movement of that chemical across the plasma membrane. Under both of these types of transport, there are subcategories. So we want to explain and give examples for those. So under passive transport, we have simple diffusion. Simple diffusion is easily demonstrated through this concept. Imagine you had a beaker of water, and imagine you dropped a sugar tablet into it. Over time, what will happen is that sugar will dissolve, and it will start to spread throughout the beaker of water. That's because on the atomic and molecular level, these chemicals are all moving around and bumping into each other, as we saw when we talked about electrolytes dissolving into water, and then over time, they spread out to be evenly distributed throughout the water. This process is called diffusion. Another example of that is movement of things like smells within the air. I give this example because it's an example where we're dealing with two gases, not just something dissolving in water. So when the gentleman here is sprayed by a skunk, what happens is the skunk's chemical goes out into the air and then it diffuses throughout the air, providing that stink everywhere that it diffuses to. An example more related to our body would be inside our lungs. Inside our lungs, we have small sacs called alveoli. They are surrounded by blood vessels. When we breathe in fresh air, we get a certain amount of oxygen in the air that goes into our alveoli and then diffuses across the wall of the alveoli, across the wall of the blood vessel, into the blood. The opposite happens with carbon dioxide. Carbon dioxide that's being produced in our body will come through the blood and then diffuse across the blood vessel into the alveoli, and then we will be able to breathe it off. This happens because each of these chemicals is going from high concentration on one side to low concentration on the other. There's no energy required to do any of these movements, and there is no carrier required in this case. Another type of passive transport is facilitated diffusion. This is very similar to simple diffusion, but if we look at that word facilitated, it means to help or to be helped. And so in this case, we're looking at a picture of the plasma membrane. We have extracellular fluid above, cytoplasm below, and then we have a molecule being transported by a carrier protein. As we mentioned when we were talking about the selectively permeable membrane, some chemicals can't move through the plasma membrane on their own because maybe they have a charge or they're too big. And so we can have carrier proteins that allow that molecule to go into it. When that happens, a chemical change happens such that the carrier protein changes shape and opens up, allowing that charged particle or that large particle to move into the cell. An example of this would be if we were trying to move sodium into the cell or potassium out of a cell. In order to do that, because the sodium is an ion that's charged, the potassium is an ion that's charged, and therefore won't go through that lipid portion of the plasma membrane, it requires a carrier protein, even if those chemicals are going from high to low concentration. Another example of not an ion would be glucose. If you recall, glucose was a fairly large molecule, C6H12O6, and so 24 atoms altogether. And so in order to move that molecule into the cell, it requires a carrier protein in order to help with that diffusion, again going from high to low. But in this case, it's still passive because it's going from high to low and because no energy, no ATP is required. Another type of passive transport is osmosis. Osmosis is actually very similar to facilitated diffusion, but it specifically has to do with water. The water would require a protein to help it move from high levels of water to low levels of water through the selectively permeable membrane. Beyond the protein helping it though, let's look at an example where we have in this case a beaker filled with water, then we have a selectively permeable membrane in the middle. On side A we have a dilute solution, and on side B we have a concentrated solution. Let's just imagine that that solute is sodium. On side A we don't have as much sodium as we do on side B. What will happen over time, as the purple arrow here indicates, is that the water will be attracted across that semi-permeable membrane from where there's a large amount of water in the dilute solution to where there's not as much water on the concentrated side. If we were to let this sit for a while, we would come back to this situation. The water has undergone osmosis to go from side A to side B until the concentrations of the two sides are the same. Another important way to understand how you might describe this is that when we talk about the dilute solution versus the concentrated solution, we would say that the side B concentrated solution has a greater osmotic pressure. There's a greater attraction of the water to that side. This is osmotic pressure. If we think back to the idea of sodium, sodium has a positive charge. The one side of water has a negative charge. And so that negative charge side is attracted towards the positively charged sodium. And so it moves from the one side to the other because of this osmotic pressure, this attractive pressure. Now let's move on to describing active transport. There's a few subcategories for active transport. The first is pumps. The major pump in our body is the sodium-potassium pump. As we mentioned earlier, in active transport, we're moving chemicals from low concentration to high concentration. Also, as is shown in the picture on the left, energy, ATP, is required. That ATP causes the change in shape of the pump in order to be able to move those chemicals across the membrane. Remember earlier in the course when we discussed sodium being the major extracellular cation and potassium being the major intracellular cation? This is where the sodium-potassium pump comes in. How do we end up with more sodium on the outside and potassium on the inside? Our bodies have this pump that allow us to move more sodium outside the cell than is inside and more potassium into the cell than is outside. The reason that we need that imbalance is because many of the functions that we mentioned that sodium and potassium play a role in, things like nerve signal conduction or muscular contraction, function because of this difference between the outside and the inside of the cell in terms of the two cations. A second form of active transport is endocytosis and exocytosis. Endocytosis. If we break down the term, endo means inside, cyto means cell, and the cis is referring to a process. So in this case, what the term means is the process of taking something inside the cell. In the image here, we have the plasma membrane with some red chemicals on the outside of the cell. We can see that the plasma membrane is moving in, and then will ultimately surround that chemical and form a vesicle. The vesicle then can move about the cytoplasm and bring that chemical to where it's needed in the cell. Another term that you sometimes hear for this process is phagocytosis. Phago meaning eating, and cytosis meaning the process of the cell doing that. We'll come across phagocytosis when we get to the immune system, because just like the endocytosis being shown here, instead of just chemicals, imagine that those red dots are a bacteria or a virus. we would have cells of our immune system that could phagocytose those bacteria, bring them into themselves in a vesicle, and then be able to move around the cell, maybe connect with one of those lysosomes that we mentioned earlier in order to break down those bacteria and rid our body of them. Exocytosis is the opposite of endocytosis. Exo meaning exiting, and so it's the process of exiting the cell. If we have something going on within the cell, perhaps our cell is producing hormones. Those hormones need to get outside of the cell into our bloodstream, for example. The vesicle that contains that chemical will move up, fuse with the plasma membrane, and will release that chemical, that hormone, out into the extracellular fluid. For the next learning outcome, explain the relationship between solute, solvent, osmotic pressure, and hydrostatic pressure, we've already discussed most of these, but I wanted to take the time to review them and then add to our understanding. A solvent is the liquid portion of a solution in which a solute is dissolved. We've talked about this in terms of if these red dots were sodium dissolved within water, or it might be glucose dissolved within water. Then the sodium or the glucose would be the solute, and the water would be the solvent. We've also discussed osmotic pressure. On the left side of the selectively permeable or semi-permeable membrane would be a low concentration of solute within the solvent, water. On the right side, we would have a higher concentration of solute within water. The purple arrow here is representing the osmotic pressure, the pulling of water towards the side where there is more solute. When we think about pressure, a memory trick as I have here is osmotic pressure is a pulling of water pressure. Something we haven't mentioned yet is once we get to this point where the water has moved across the membrane to the right-hand side, there's another type of pressure that's involved here. And this is the pressure of the water pushing up against that wall because the right side is higher than the left side. Or if we were talking on a biological level, because there is more fluid on the right side than the left side. This is hydrostatic pressure. This is a pushing pressure. So the water in this case is pushing against that selectively permeable membrane, creating this other type of pressure. Perhaps some of that water, because of this hydrostatic pressure, might move back through the selectively permeable membrane. We need to look at these relationships in order to summarize them overall, but also to add to our membrane transport mechanisms. There's a fourth passive transport mechanism, filtration, that we need to look at. Filtration involves this force of hydrostatic pressure. Everything else that we discussed was moving molecules from high concentration to low concentration. But filtration is dealing with moving chemicals from high hydrostatic pressure to low hydrostatic pressure. An example of that would be if we had a syringe here. We put a force on the plunger of the syringe, and then water would come out the needle of the syringe. In this case, if we had holes within the syringe and we put a force on the plunger, water would come out of the various holes that have been punctured into the needle, as well as the end of the needle. If we were to add more force to the plunger, then more water would be squeezed out of the needle at a higher rate. The second syringe here is related to blood traveling through our capillaries. These small blood vessels, as blood passes through them, have all this force from the heart pushing the blood through those capillaries. As the blood is pushing through, there's lots of hydrostatic pressure pushing up against the wall of those capillaries. Large things like the red blood cells that you see in the image here, or there are also proteins within our blood that are too large to move out of the capillary, will stay within the capillary while water is squeezed out through the spaces found between the cells of the capillary. This process is filtration because just water and other small particles are going out of the capillary into the tissue while the larger particles stay behind. Now we want to look at a few other definitions differentiating between these terms here, but so that we can better understand and pull together how all these concepts work and relate to the effect on body cells. So here we have a beaker with again solvent in it and then solute. Imagine we were to put a cell into that solution. The solution is being described here as normal saline. What that means by normal is that it has the same concentration of solute as is found inside the cell in the cytoplasm. What we would call this then is an isotonic solution. Iso means same. And tonic is referring to the tonicity of solution, which means the concentration of solution. What this term means then is that you have the same concentration of solute inside and outside of the cell. What the arrows are indicating in this picture is that the osmotic pressure on both sides of the cell is equal. So equal amounts of water will come out of the cell as will go into the cell. In this example, we're looking at a cell that has been placed in pure water. In that case, the solution is hypotonic relative to the cell, because hypo means low, and tonic again means concentration. So the water has a low concentration relative to the concentration of the cytoplasm. As the image is showing, there is a higher draw of water into the cell because of the osmotic pressure produced by the solutes within the cell. Water will flow from the hypotonic solution to the inside of the cell, eventually potentially causing the cell to break. The last type of solution here is if we had a concentrated salt solution that we put the cell within. This is a high amount of salt within that water in the solution, and so we call it hypertonic because hyper means high, and tonic again refers to the concentration. When we put the cell in that high concentration, that hypertonic solution, water would be drawn outside of the cell into the solution because of the osmotic pressure pulling the water outside of the cell. This would cause the cell to shrivel. So now we want to define osmolality. Osmolality is the pressure of a solution, so related to the osmotic pressure that we've been discussing. Osmolality, in a way, describes how thick the solution is. It's the total concentration of all solute particles in a solution. So in our normal blood plasma, the osmolality ranges between 275 to 295 milliosmoles per kilogram. Not that you need to memorize that value, but it's a value we can actually measure about how concentrated something like the blood is, and can be helpful in terms of diagnosing a patient, analyzing things like dehydration, and so on.